session. My name is Jeroen Aerts, and I'm a customer technology advisor at Resolve Biosciences. So at Resolve Biosciences, we're developing a highly sensitive, high-resolution spatial transcriptomics technology called Molecular Cartography. And you can already see in the background a nice teaser image of a cross-section through the human gum. And each individual spot that you see here is an individual transcript. So this already tells you something about the resolution that we offer. And it also, of course, generates beautiful images, I think. So it's great to see the community here in spatial biology coming together and showcasing many amazing applications for spatial biology. And given the session today, I want to highlight some of those applications where molecular cartography allows or enables researchers to make new discoveries across different fields within a pharmaceutical context more, given the topic of this session. And I will highlight some of the applications here where molecular cartography can be used, for example, for biomarker discovery or toxicology, tissue atlasing, of course, but many more as well. And I think spatial biology in general can be applied across life sciences with many great applications. But of course, to enable researchers to make these new discoveries across these different applications, we of course need to allow or enable our platform to be compatible with many different tissue types. And molecular cartography is very flexible in this case. So we made our platform compatible with many different tissue types, and this is just a glimpse of some of the data sets that we produced. So mainly focused on mammalian tissues here. So this is mouse and human data, for example, for liver, ileum, kidney, and beyond. And we have a dedicated team at Resolve Biosciences really optimizing protocols for new tissue types, and we continuously expand on this. So we have now more than 20 protocols available, but at Resolve Biosciences we also realize that you have many spatial biology questions going beyond the mammalian tissues. And so at Resolve Biosciences we use molecular cartography also to answer spatial questions for many different tissue types going beyond the mammalian questions. So here you can also see some nice data sets on, of course, important sample types like organoids, embryonic samples, and of course it's compatible with cell culture. But we go way beyond that. So we have protocols available for Drosophila, zebrafish, even Xenopus. And I'm proud to say also we are heavily invested in the ag bio industry, showcasing some nice spatial data sets here for maize in this case. So we have a lot of experience with plant tissues as well. So how do we generate these data sets? So how does our technology work? So molecular cartography is a form of combinatorial single molecule fish or barcoded fish. And so this is an overview on the left side of a basic protocol where we provide glass slides. These are microscopy-sized glass slides, so basic glass slides with 8 different positions, 8 different wells where you can place your tissue section, be it cryosections or FFPE. And then we synthesize transcript-specific probes against 100 targets per sample. And in a single overnight hybridization step, we hybridize all those probe sets to the target RNA. And then we have a patented chemistry where we're able to colorize all these transcript-specific probes, but also decolorize them. And this allows us to go in a cyclical imaging approach, really to increase the multiplexing capacity of our technology, but still adhering to the single molecule fish principle. And that is illustrated on the right. So in the way we design our probes, we know upfront by our in silico design that, for example, for transcript A, in this case coming from gene A, we know upfront that in the first round of imaging, we expect it to light up in one of the 2 channels. So we use 2 channels and 8 rounds of imaging. Then we decolorize the sample, go in with another colorization round, and again, we expect it to light up in the same channel or in the other channel, depending on in which round we are imaging in. And going through all these different rounds, you see that for each individual transcript species, we are basically creating a unique barcode. And this is the principle of barcoded fish, where at the very end, you can decode these barcodes, and then in the spots in space that you were tracking, annotate that spot with a transcript identifier. And at that moment, you have completely digitally quantifiable data. So you don't need any fluorescent intensity or optical density to measure differences in your sample. So this is a very elegant way of quantifying data, and you can actually do transcript counting with this type of technology. So we've seen many great technologies yesterday, today as well, for many great applications. So it's important to know what molecular cartography can offer for your research. So one of the important aspects of our technology is that we don't use any clearing methods or proteinase K, so the sample remains completely intact. So this allows you to go in afterwards, after capturing the transcriptomic data, using immunohistochemistry to detect proteins, or even doing some DNA analysis afterwards. We currently offer 100 genes per sample, but we do have a roadmap where we aim to go to 300 genes per sample. I think we're the only technology in the space today that captures the full 3D information in a 10 micrometer sample, where we offer a single molecule resolution in the XYZ position. And this allows us also to increase the spot calling accuracy and have or offer a very high sensitivity, so close to what you will achieve with single molecule FISH, with a very low false positive rate as well. And as mentioned before, one spot is one transcript, so it allows you to digitally quantify all the different transcripts. And of course, tissue plexity, sample flexibility is really what our platform enables. And just to showcase what it means to have subcellular resolution in the 3 dimensions, on the right side you see here an overview of a hemisphere of the mouse brain. Using molecular cartography, you can zoom in down to the single molecule level and show all the individual spots fully in 3D. And this is an example of a single interneuron, so a somatostatin interneuron, where each individual yellow spot here is an individual RNA molecule, somatostatin RNA molecule, and then surrounded by targets for 99 additional genes. So here you can nicely see that we do capture the full 3D information, and this also allows you, if you have questions, for example, related to the different distributions of transcripts within a given cell and how those compare to other cell types or between samples, this is definitely possible with our technology because we offer that subcellular single molecule resolution. So I'll demonstrate some use cases specific for certain sample types on how you can use our technology in spatial biology in general to answer important questions in the fields of biomarker validation, TITV, and much more. So starting off with the brain, I want to highlight a certain study that we did with a pharmaceutical partner, and they were interested in disease-associated microglia. So these are specialized immune cells within the brain that are present in Alzheimer's disease and are closely related to the amyloid plaque pathology. So what we did in the study, we compared wild type to a diseased mouse model, specifically in the cortex. And as you can see here, just highlighting the major cell types, neurons, microglia, etc., you can already see major differences between the wild type condition and the diseased condition. So much less neurons and also an increase in the number of microglia, especially surrounding the amyloid plaques. But this is also showcasing you that after molecular cartography, we actually stained these tissue sections with PFTAA to label the amyloid plaques. So here in yellow, you can nicely see these amyloid plaques only in the diseased condition. And some of the types of analysis that you can do is just doing differential gene expression analysis and showcasing which transcripts are upregulated, but especially where within the spatial context here. And then you can see that we see these enriched transcripts surrounding the amyloid pathology. So this is again a demonstration of the power of spatial biology. And what else can you do with this type of data? Well, you can actually extract single cell data from this. So you can segment each individual cell, extract that single cell data and plot it in a UMAP plot like you see here. So very reminiscent of what we know from single cell RNA-seq. And then you can of course compare the wild type and disease conditions. So this is This was already known from 2016-17, where the first publications came out identifying these disease-associated microglia. So specifically in the diseased condition, you can see a unique population of microglia here. But for the first time now, we can use the spatial component and overlay that in this data set. So when we measured the distance towards the center of the plaque, you can see that the closer you get to the center of the plaque, the more you get into these disease-associated microglial populations. And this is just plotted on a UMAP plot. You don't really have the spatial insight here, but you can always go back to the original data, and this is just showcasing some of this analysis that was done. So we analyzed more than 600 individual plaque niches, and we drew concentric circles around these plaques and counted individual transcripts. And as you can see, the closer you get to the center of the amyloid plaque, the more counts you get for these disease-associated microglial markers. You can think about this in other ways as well. You can include different genes related to certain pathways, and you can start doing pathway analysis in a spatial context. And on the right, you can see that you can also zoom out and do this on a single cell level. So counting the number of cells closer to the amyloid plaques, you can actually see an increase in the number of microglia and a decrease in the number of neurons. So again, demonstrating that spatial biology can really uncover novel insights and can be a very powerful tool in drug discovery, biomarker validation, etc. So with the same pharmaceutical partner, we're also looking into human samples. So these are human post-mortem brain samples, fresh frozen, control versus Braak stage 6 Alzheimer's disease samples. And as you can see, there's major differences between these samples, and we're continuing to explore these differences and really highlight potential druggable targets in these samples. So, but this is an ongoing study. And I want to move on to a next important tissue type, which is the liver, which is incredibly rich in spatial data, of course. And you can use, or I will show you some applications where you can use molecular cartography in the context of atlasing and biomarker discovery. So here you can see an example of a mouse liver. And in the sea of red, which are basically the hepatocytes, when you actually zoom in, where we offer the single molecule resolution, it's quite easy to identify, just based on a couple of markers, these immune cell types like Kupffer cells and T cells in blue. And as you can see here, when you zoom in on the central vein, you can actually quite nicely see the single cell layer here identified by VWF as endothelial cells lining the central vein, so the blood vessel. And then the portal vein, here you can actually see very nicely these cholangiocytes in green that produce bile in the bile duct. So our technology offers you applications in terms of atlasing and really looking into very rare cell types, offering you high resolution. Also to showcase in the same data set, you can actually zoom in onto these zonation patterns that we see in the liver. And actually zooming in on these zonation patterns, you can actually use the single molecule resolution to really go in quite deep in your analysis and see these different gradients, in this case, for example, for CYP2E1, and really map out these gradients and quantify these gradients that make up this zonation in the liver. And so again, similar to what I showed you for the brain example here, you can actually extract the single cells, do clustering. We identified in this data set 7 different subtypes of hepatocytes, and we mapped them back onto the tissue. And you can actually see in this beautiful image, again on a single cell level, these beautiful zonation patterns going from the central vein all the way to the portal vein. And you can actually use zonation in other contexts. And to showcase this is unpublished data from a customer where they were interested in tracking zonation in a spatial context, of course, in relation to toxicology, really to a certain exposure to a certain toxicant. And as you can see here, when we label the different, or when we visualize the different markers for the portal vein, central vein, and also the cholangiocytes, you can actually see when these mice were exposed to increasing concentrations of toxicant, you can actually see a loss of zonation. So this demonstrates that in toxicology as well, there are many different interesting applications for spatial biology. This brings me also to a recent study that was published using our technology. So this is work from Martin Gilliams from the VIB in Ghent in Belgium. And so this is both from Martin Gilliams and Charlotte Scott. So they did a huge effort in mapping the spatial proteogenomics landscape of both mouse and human liver. So they're single cell experts, liver experts, and they developed many different protocols to extract all different cell types from mouse and human liver, and they combine that with single cell RNA-seq, single nuclei RNA-seq, and also combine that with site-seq with over 161 antibodies included, really to generate and complete single cell atlas of both mouse and human liver. And they combine that with Visium to really have a broad view over the entire tissue section and extract additional spatial information. And using the combination of single cell RNA-seq, single nuclei RNA-seq, and Visium, they curated a highly specialized gene list to really go in deep with molecular cartography to identify very rare dendritic cells. As you can see here in blue and pinkish color, you can actually quite easily pick up all these transcripts for these very rare dendritic cells, and they were not able to capture these very rare cell types with either Visium, but also not with a highly multiplexed protein spatial technology, because they could not get these antibodies optimized. Of course, both technologies have their place in the spatial biology context. They're very powerful, but this is a great example of how you should think about molecular cartography in an atlasing effort. So this really showcases the power of highly sensitive and high resolution spatial transcriptomics. So I want to end with some other examples within the context of oncology. So first of all, we have a lot of experience in the oncology field with many different sample types and protocols optimized for this, showcasing some examples in this case for human breast cancer, where just highlighting 14 different genes, you can actually see major differences between the two different patients. And then you can, of course, think about differences in the tumor microenvironment, really dissecting the difference between the two patients in the context of patient stratification. And I also want to highlight another study we are doing with VIB. So this comes from the lab of Chris Marine in Leuven. So this is unpublished data, and he's really interested in tracking different melanoma cell states related to the spatial context of the blood vessels. So really tracking the tumor progression. And this is showcasing some of the data where he zoomed in on certain cell types. This is mouse skin, and the next steps in the study will also be looking at human and FFPE material. And here you can see the closer you are to the blood vessel, the earlier you are in the metastatic process of these melanoma cells. So the further away you go from the blood vessel, you see that these cells become melanocytic, and deeper into the tissue you come in these hypoxic regions. So this showcases again the power of spatial biology, really enabling you to track these individual cell types in a spatial context and gain novel insights with this. So this is a very interesting ongoing study. And I want to go further. So how to access our technology. So we have several workflows in the field. So our workflow consists of an imager, a robotic arm, and a liquid handler. So this is a completely walk-away solution. So you do sample preparation, you load in the samples, and you load in the reagents. And then it's a walk-away solution. It's fully automated, and the data will be generated in a few days. And on the other side, you have the end-to-end service, where we have a probe design tool online where you can design your own probes in silico. And we synthesize the probes. We send you glass slides where you can mount the sections you make. You send them back to us on dry ice, and then we do everything else. So we run the full molecular cartography chemistry, and we do the primary analysis and also part secondary analysis. And then we schedule a meeting together with you to go over all the QC metrics and really discuss the data together with you. And we also offer additional support in terms of analysis. We have a dedicated bioinformatics team that is available to help you out in any other analysis questions you have. So in terms of output, what kind of data is actually generated? Because we are using a barcoded fish approach, you're not dealing with terabytes of data that you need to manipulate and really visualize to extract the information. So we actually generate count matrices with X, Y, Z coordinates and a transcript identifier. So if you detect, let's say, 2 million individual spots in your region of interest, you basically have a count matrix with 2 million individual rows with the coordinates and the transcript identifier. We offer you DAPI images, raw images, and also bright field images. And of course, as mentioned before, we generate a full data report where you can go over all the QC metrics we check in terms of total number of transcripts, false positive rates, interesting spatial distributed transcripts, and also some quality cutoffs we employ. So this is part of the complete package. And we also offer different analysis tools. So on the one hand, we have Polylux, and this is basically a plugin of ImageJ. So this is designed to be very open. So the count matrix that I showed you just now is basically a text file, and you drag it into the plugin of ImageJ. All the genes that you selected in your gene list will appear. You can give them any color that you like. You hit the update button, and it will visualize all the different transcripts onto the DAPI image or bright field image that you loaded into ImageJ. And then you can use the power of ImageJ to your advantage. So you can really import single cell segmentation ROIs. You can export that in a cell to gene matrix again, and make the connection to tools like Seurat to do single cell clustering. So it's really an open design. It's compatible with any data format that you can imagine. And it can easily make the connection between other open source tools for cell segmentation and also single cell clustering. On the other hand, we also offer a web-based tool which is called Recognize, and it's basically a visualization tool where you can visualize all the different transcripts of the genes in your gene list and really fly through the tissue fully in 3D. And it has Seurat running in the background. So this is really when you import some single cell RI segmentation data, you can easily generate these 3D UMAP clusters and select one of these clusters, for example, and then map those clusters back onto the tissue. So this is a more streamlined type of visualization and data analysis let's say, in coordination with Polylux. So Polylux offers you the flexibility in going really deep in your analysis, and recognizes really to visualize and make nice pictures for publications, for example. So just to end, how can you think about molecular cartography in, I would say, a competitive landscape context? Because we have, of course, many different technologies that are presented today. Resolve Biosciences or molecular cartography is really the high resolution, high sensitivity technology, as opposed to some other technologies that are more sequencing based. They have other advantages like very big field of view and throughput, but of course they lack the sensitivity and the resolution. The same goes for amplification based FISH methodologies. When you amplify these signals through rolling circle amplification, it's an enzymatic process, it's not 100% efficient. So you will lose on sensitivity as well there. So we stick to the original single molecule FISH chemistry, allowing to have the highest sensitivity and highest resolution in that field. So this is how you need to think about molecular cartography. So I want to end here, so please come visit us at our booth and meet my colleagues Tina, Karan, and Justin that are here today. So we're happy to answer all your questions at the booth, really enable you to discover new things using our technology, and we're all very passionate about spatial biology. So feel free to come talk to us. We're always happy to get to know you. Thanks all.
Great, thanks Jeroen. So we do now have time for Q&A. As always, we do have a mic in the middle, or I can run around to a mic. We do have some questions on Slido, so I can kick off with those. So is your technology compatible with endogenous reporters such as GFP?
Good question. Yeah, that's definitely the case. So GFP is compatible, so it does not interfere with the imaging channels that we use. Of course, you need to always be considerate of what's the use of GFP in your model, because during sample preparation it can happen that your GFP signal gets quenched, but we can also incorporate GFP probes in one of, so in your gene list you can incorporate GFP and then visualize GFP simultaneously in your sample, or you can stain it afterwards with immunohistochemistry. So we offer a lot of flexibility there, but GFP in general is not an issue and it will not interfere with our assay.
Great, thank you. And then do you offer support in data analysis?
Yes, so as mentioned before, we have a dedicated bioinformatics team, and we have many cases where customers want to have additional support in cell segmentation, which of course is a very big challenge in spatial biology in general. But we have a lot of experience with the tracking, let's say, the new tools coming out from the community for single cell segmentation. And we have a lot of experience in developing these machine learning algorithms in combination with membrane markers or other metrics in our spatial data. So we have, for example, great success with machine learning algorithms for segmentation for human and mouse heart samples and for many other sample types as well.
Great. And then last question on Slido. How long does it take to get data in the end-to-end service option?
So generally, when we start synthesizing the probe sets all the way up until we, let's say, schedule a meeting with you and deliver you the data with the data report included, this takes around 6 to 8 weeks to completely finish that project.
Great, we've just had a new one through as well. So is this technology agnostic, and can the data be analyzed in different platforms?
Good question. I'm not sure what agnostic means in this context, but you could in theory analyze the data in any other tool. I mean, the tools we provide are just helping researchers, just the data that is retrieved from the instrument, to go ahead and use our tools. But we have many customers that actually don't use our tools and have other spatial analysis pipelines in place. They just take the count matrix, which is basically a text file, and they manipulate it in any way that they want. So that is also, for us, I think, an important feature. We want to enable the community to use our data, and that's why we just have a very open data format.
Brilliant.
I was interested, is that on? So I was interested in the 3D aspect.
Yeah.
Can you tell me how thick the sections can be and what the resolution actually is in the Z?
Yeah, good question. So we, for now, are limited, or I would say the feature we have is 10 micrometers. And this has to do with how our optical setup is present in our workflow. So it's the 10 micrometers, and then we take throughout the full 10 micrometers all the Z stacks necessary to resolve those single molecules. So we have the same resolution, 270 nanometers XY, but also in the Z axis. So that allows you to also in the Z axis determine each individual molecule and resolve the full data volume like that. It's diffraction limited imaging.
Great. How large is the area of tissue stained on your slides?
So when you consider we have a glass slide with 8 different positions, so each of these positions on the glass slide where you can place your sample is a square centimeter. That is basically where you can place your samples. You can have one big sample or just many small samples into one well. And then you can actually select within those 8 different wells the region where you want to capture data. And so the full region where per glass slide that you can capture is around 26 square millimeters. So this has to do with our imaging throughput. So we are really, as mentioned before, capturing the full 3D volume. It takes a long imaging time, but it offers you the highest resolution and highest sensitivity with our assay. And that's why we limit the field of view to around 26 square millimeters.
Great. Any other questions?
Can you access the data in the long term? For example, if you're interested in a region and then, I don't know, 1 year later, you want to come back to the slide?
Yeah, of course. So all the raw data is, of course, stored also in the external workflows we offer. So once you have the count matrix, these are typically per experiment, I would say 10 gigabytes, so quite easily transferable and shareable with other people. But you can always go back to the raw images stored, let's say, more on a passive storage. So these are, let's say, the raw imaging data with terabytes of data, basically. But you don't need to access them all the time, only when you want to go back and rerun the primary analysis, if that is necessary. But once that primary analysis is successful, which is almost always the case, of course, once you have that count matrix, you can always access that, and it won't change, basically. So you can share that quite easily with many people and go back at any time.
Thank you.
Any other questions? No? Well, thank you, Jeroen.
Okay.
Great presentation.